Okay. Recording.
Okay. Well, thank you. Hopefully you'll stay with us, Kevin. That's
Yeah. Becca, do you want to try sharing screen and pulling up the PowerPoint?
Yeah, give me 1 second to download it real quick.
Make sure you have the video ready to go too.
Yeah, I will do that. Just give me 1 second. I will start sharing the PowerPoint and then I will drop the video.
perfect. So you're set, Becca?
I think so.
Okay, great.
Oh,
Well,
wait.
good morning. Good morning to everyone. And thank you all for getting involved this year by volunteering in the BPC Citizen Science Program. It's really critically important that we do the work that we're doing. And so thank you very much. I'm really excited about 2022. This is our sixth
Okay,
year.
I'm okay.
If everyone could just mute their microphone. So 2022 is our sixth year of sampling and testing the ponds in Brewster with our partners at APCC and the University of New Hampshire and LimTech. The program has grown from its beginning in 2017 when Cameron Malkus Benjamin and Chuck Medansky really started the program and kicked off citizen science at BPC. And that year in 17, they tested 4 ponds and had 5 or 6 volunteers. So we really are off and running. And so today in this season, we will have 19 ponds in our regular rotation. All 6 town landing ponds in Brewster will be in that 19 pond group. So we're helping the town by doing that, of course. We'll have over 50 volunteers this season. And what's really exciting about it is we have 80% of the volunteers who participated last year are back as citizen scientists this year. And we have 12 new citizen scientists this year. And all of those individuals that are new will be paired up with people who have done it many, many times, some right from the beginning. So it's great to have 12 new people. So a particular thank you to the new people getting involved this year. And we hope that you stick with us for a while. Citizen science really is, I could wax about it, but it's really critically important because if we didn't have you guys, this work just wouldn't get done. We have we live in a town where the staff is the administrator, the Department of Natural Resources, our health agent in town are really, really invested in our freshwater ecology and taking care of it. And so that's really, really positive. And citizen science helps to that end as we are, in addition to the town staff, we're helping to educate and make the citizenry in town really aware of what's going on. And, by participating in this kind of program, you really become a stakeholder. And you'll look at a pond, every time you go to your pond or any pond, you'll look at them all differently going forward. And I think that's a great benefit of citizen science as well. So I would just say, if you guys would talk to your neighbors and your pond groups and let them know what you're doing, why you're doing it, and encourage them to join BPC if they're not a member. So that's a little bit of sort of an overview of why I think citizen science is important. There just aren't enough scientists to do the work that we all do. And so thank you for helping to continue, I think, BPC's cornerstone program that, again, was started by Karen and Chuck. During the season, what I'll do is I'll communicate the test results to you guys every other week. They come to me from Becca and from Kevin, the results of the analysis in the lab. And I'll communicate first to the citizen science team, next to all the BPC subscribers, and we have 1,000 or so. And then, of course, simultaneous, we'll be talking and communicating the results to the town administrator in Brewster, the Department of Natural Resources, and our health agents. So, being an ally and a partner with the town is really important to us. So So today's meeting, and here you can see the picture of, I think it's, what pond is that, Becca?
It's a sheep pond.
I thought it was sheep pond.
From
Yeah.
last year. Yep.
Great. Okay. So beautiful picture of sheep pond. We'll keep the agenda at 60 minutes. Kevin will talk about the science of cyanobacteria and overview. I'll give you a little bit of a 2021 year overview. Becca Miller will talk about monitoring protocol, and she'll show her great sampling training video of actual collection. Then I'll go over the teams and the schedules for the season. And then we'll open it up for questions that you guys might have. So Kevin, I'll turn it over to you.
Awesome. Well, thank you, Marty, and thank you all for being here and for being a part of this program. It's been a long partnership between APCC and BPC that has ensured that we've been able to gather a tremendous amount of data from our Brewster ponds, and it's tremendous to work with a group like BPC, and a town that's passionate about their freshwater ecology. Becca, if I do drop off, I'll share it over. I'll pass it over to you to talk about these slides. But I'm feeling a little better about my connection at this point. Just sorry again for the technical difficulties on my end, but looks like we're going to be off and running. Just want to note too that this, that for the 80% of you that have been doing this since last year, or for a couple years with us now, some of this will be review. But we do have some updates to our program this year, so I do encourage anybody who is going to be monitoring with us this season who isn't a part of this video to tune in and watch the recorded copy that Becca will send over to Marty. So with that, yeah, I'll talk about an overview of cyanobacteria and our plan for the season and whatnot before passing it back to Marty. So cyanobacteria are common aquatic microorganisms, not just in our freshwater ponds here on Cape Cod, but in freshwater systems across the globe. We also find them in marine systems as well. They're a very large group, and there's many groups of cyanobacteria that are believed to be harmless to humans and wildlife. But what we do in this program is focus on the subset of cyanobacteria that we know to be potentially harmful to humans and to the environment. And two of those I've included in the picture up in the top right here. That's Dolichospermum and Microcystis. We see these most common, on our ponds on the Cape. We see both of them in Brewster, Dolichospermum in particular. Dolichospermum is the one that looks like a green beaded necklace, and Microcystis is the one that looks like a bunch of dots all accumulated together. And these are both known as potentially able to produce toxins. A key part of our program is that we're not just looking for presence or absence of these toxin-producing cyanobacteria. We expect to see toxin-producing cyanobacteria in all of our ponds. Even in the most untouched remote ponds on the Cape, we still see these toxin-producing cyanobacteria, and in their normal concentrations, they present no to very little potential for harm to humans or wildlife. But of course, it's when they grow out of control, tend to dominate their pond ecosystem, that we become concerned. And at these points, you might see them accumulate into what I have on the bottom right here, which is what we refer to as a cyanobacteria bloom accumulation. These clusters of cyanobacteria can sometimes be visible to the naked eye. You might see a little green dot, green dot kind of on the surface of your ponds, but it might not have, it might have accumulated a much of these little green specks, to create, a harmful cyanobacteria bloom. But of course, when we do have an accumulation like you see in the bottom right, that biomass of cyanobacteria is what we become concerned with and is the level that we want to take action on. And so some other notes about cyanobacteria, some of them are nitrogen fixing, like Dolichospermum here, which gives it advantages in certain systems. There can also be buoyancy control among some groups, able to control their ability to, perhaps start on the bottom and then, make their way to the surface to collect sunlight and so on. And yeah, as I said, it's the toxins produced by cyanobacteria in particular, the quantity of these toxins that we're concerned with. And when I refer to toxins produced by cyanobacteria, I'll use the term cyanotoxins interchangeably. And as you all have worked in the past with Nancy Leland on her food web studies, looking at cyanobacteria and the larger kind of pond ecosystem food web and the potential connections there, we are concerned with the effects of cyanobacteria on pond ecology as well. And that's something that's been a great benefit for us, to work with BPC and Nancy and Jim on their work to learn a bit more about how cyanobacteria are affecting other areas of our freshwater ponds other than the human public health impacts. So you can go next slide, Becca. All right, so just to cover the two kind of big areas of why we monitor cyanobacteria, as I said, the first I'll go over is the public health implications and our concerns with those cyanotoxins. You can be exposed to these toxins produced by cyanobacteria. First off, I'll say skin contact. There's dermatoxins, which can be skin irritants. Where you go swimming in your pond, you come out, you may have a rash if there were those toxins in that pond. That can often be the extent of our concerns with skin contact to cyanotoxins. There's also inhalation of toxins in water vapor. Just again, I want to remind everyone to mute their microphone if they can. Yeah, the inhalation of water vapor that might contain cyanotoxins, as you all have been working, as I said, on Jim Haney's project with aerosolized cyanobacteria. This has been a focus there. Of course, there's been documented cases of respiratory irritation, other issues from cyanobacteria that we're hoping to learn more about. Yeah, here it sounds like there's some feedback. I'll see if I can mute temporarily. All right.
I think I muted them for you, Kevin.
Okay, thank you. Yep, I think we should be all set now. All right, just getting back to it. Other areas of exposure can be consumption of cyanotoxins. This is one of our, for the standards that we have from Mass Department of Public Health for cyanobacteria exposure, one we're particularly concerned about. Toxins produced by cyanobacteria can include hepatotoxins, which can cause liver damage or other organ damage. Consumption of pond water can often be the cause of pet deaths from exposure to cyanobacteria toxins. As you know, as they're going by the ponds, they may directly consume pond water or go swimming and then groom themselves afterwards and accidentally consume that pond water containing cyanobacteria toxins. So that is a concerning direct avenue for cyanotoxin issues. Of course, we're also worried about human impacts from consumption of pond water containing cyanobacteria toxins, and this can occur during swimming. Children in particular are a bit more at risk since they tend to consume more pond water as they swim than adults, and also with lower body masses can also be more at risk. And as I said, with I have a bioaccumulation there, but I might remove that and just keep it as exposure to contact, inhalation, and consumption. I think perhaps I had that note there, as there have been some studies on cyanobacteria toxins in freshwater fish, and we have concerns about consuming those fish, that might contain those cyanobacteria toxins. So moving on to pond impairment, the other main reason why we monitor for cyanobacteria. Cyanobacteria thrive under conditions of high nutrients and warming temperatures. When I refer to nutrients, I'm mostly referring to nitrogen and phosphorus enrichment. By studying cyanobacteria trends in our freshwater ponds, we can get a better idea for which ponds are potentially have higher nutrient loads, or are just more vulnerable to cyanobacteria risks. And over time, the trend that we see with pond impairment with these impacts is the ongoing process of eutrophication in our freshwater ponds, which is the degradation of our pond health. As you have increases in nutrients and warming temperatures, this favors the cyanobacteria and other algae and phytoplankton in the ponds. This can cloud the ponds and decrease clarity. All of this biomass, can die off over time and descend to the bottom. And without sunlight able to penetrate the bottom, the bacteria will break down that organic material in absence of sunlight, which will use up all the oxygen at the bottom of the ponds. This can stress out anything that lives at the bottom, any of our freshwater fish. And we often see fish kills associated with cyanobacteria and other eutrophication issues due to these low oxygen levels. Of course, cyanotoxins can also cause fish kills as well, but typically, oxygen stress can be a main driver. And through all of this can lead to a decrease.
Sorry, Kevin, I accidentally muted you.
Yeah,
I was trying to hit the
no
mute
worries.
person.
And lastly, I'll just note that by studying pond impairment and which ponds are more vulnerable to cyanobacteria risks, towns can then focus more attention to which ponds are having these yearly cyanobacteria issues to try to target their nutrient enrichment to try to improve the health of those ponds. So you can click next slide, Becca. All right, so here at APCC, we work with groups like yours across the Cape. Of course, BPC is kind of where it all started for this program, and it's the work that we've done with BPC has helped inform the work that we do in other towns, this partnership has helped this work grow across the Cape. And increase our ability to fill in monitoring gaps for cyanobacteria in areas where it likely otherwise wouldn't happen. We work with towns like the Brewster Health Department to keep them informed of cyanobacteria risks so that they can promote recreational safety. They can post advisories like the one that you see here on the left, and to inform the public of when they should stay away from a pond, and likewise when it's safe again to return to interacting with that pond as normal. And by doing all this, we hope to raise awareness. We hope to raise awareness of what cyanobacteria issues, what issues there are, what the impacts can be from being exposed to cyanobacteria, what cyanobacteria blooms look like, so people can inform us of when they might see a cyanobacteria bloom that we can then test. They can understand, just as I said, just that in many of these cases, it's just going to be, say, 2 weeks of staying away from your pond, and then it's normal to go back and interact with your pond again. So understanding all those areas at play, and then also the nutrient side of identifying, these vulnerable ponds and having folks trying to reduce their nutrient impacts on their ponds to try to motivate action to improve the quality of our ponds and pond health in each of our towns. All right, next slide. All right, and before I turn it over to Marty, just wanted to give a quick look at our plan for the entirety of the Cape this year. As I said, we've had a couple updates to our program, but starting off, as we did last year, we will continue to monitor in all 15 towns on the Cape, about 140 ponds in total on a biweekly basis, including our, I believe, 19 ponds in Brewster. The Barnstable County Department of Health and the Environment, or the BCDHE, their water quality lab this year will be able to do confirmatory toxin testing on our samples, which is a huge and great step forward in cyanobacteria work on the Cape. They'll be able to take precise toxin measurements on samples that are deemed necessary for this additional testing. This testing is expensive and requires requires expensive equipment, and it's a bit complicated as well. So using a program like ours to flag samples for this additional analysis works perfectly in tandem with what the county is getting ready to do, and this will be a great partnership going forward to increase the data and understanding of toxin risks that we have in our ponds, to be able to give health agents like Amy Von Hohn in Brewster here an increased ability and confidence to determine what the risks are to promote public health. And as Becca will go over and Marty will touch on too, there will be an additional sample we will ask you all to take this year. It's a very easy sample to take. I'll say it's the easiest sample we will be asking you to take. I'll leave it to them to discuss that sample, but that is an update to this year. Another update is that we are working even closer this year with the Mass Department of Public Health on receiving guidance for cyanobacteria scums. We will pass on the photos that say Chuck takes and Cliff, or that any of the rest of you take of cyanobacteria scums, to get guidance to have the Department of Public Health let us know of at what level of or size of those cyanobacteria scums they would recommend an advisory, and once again make things simpler and easier for each of our town health agents, and to give them more data and more guidance to work off of. And we've also been taking the off season to work with the Cape and Islands Health Agent Coalition. You've had great representation in those meetings by your own Marty Burke, to help give insight to the health agents on the Pond Coalition and citizen scientist aspect of this work. And we're looking to continue to have these conversations with our health agent partners to increase the understanding of cyanobacteria work done here in the Cape, and how we can continue to tweak what we do to serve our health agent partners all the better. So with that, I'll turn it over to Marty to speak on sample collection, but first review our results from last year.
Okay, can you hear me? All right,
yep.
Terrific. Wasn't sure. That was a great presentation, Kevin, and I think the confirmatory toxin testing and the work done with the health agents really over the last 8 or 9 months with APCC and others has really brought this effort to a full transparent position where we're all talking to each other and we're trying to understand how to best serve the public. And the reason why is what Kevin hit on earlier, it's really all about public health and pond impairment, but primarily it's public health. So here I just put together a quick overview because I think it's nice to tell you all what happened last year. And all of this information is available, by the way, on the BPC website. Complete year-end reports I've put up really over the last, I think, at least 3 years, including a citizen science overview piece. And of course, as someone said earlier, we've all said this training will be recorded and put on the website as well. But last year, the season began May 24th, ended November 2nd. And at last year, we had 17 ponds in our regular rotation, including the 6 Brewster Town Landing ponds. We test bi-weekly. There were 5 ponds that were brought to our attention. This is the whole awareness of the citizenry. People see what we're doing, hear what we're doing, understand what we're doing. They may call me up or sometimes call Becca or Kevin up and say, I saw something funny at XYZ pond that's not on the rotation. Could you take a look at it? And we offered that service and gone out. Either I've gone out or Becca's gone out, and we've grabbed samples, usually same days we get that call, and tested 5 additional ponds. So that's still something that can happen. We're about 50 scientists, volunteers last year, and 325 or so, don't quote me on that, but it was a big number. Pond water samples were collected and tested. The data gathering during the sampling is really the data that you're going to be capturing on the raw data sheet. The volunteer with the clipboard who's filling in the written information will be gathering data on the current weather conditions, water, pond observations, turbidity readings. Of course, then the samples will be collected. That'll be a whole lake water sample, a BFC sample, a surface grab sample. And then if there is a scum that you see, a visible scum that may look like spilled green or blue paint, you would grab the scum sample and label that accordingly. And then next slide, Becca. At least I don't see everything on the same slide. Right, thank you. So this is important too. The postings in 2021 in the town of Brewster were at these 5 ponds. And Cliff Pond is in Nickerson State Park. That's Chuck Medansky's personal pond, and he's the one who's collected the samples and tested the samples since 2017 at that pond. And the posting is done by, I put Mass DPH, but it's also a partnership with Mass Department of Conservation and Recreation, so Mass DCR, that was posted on June 11th and October 11th. Lower Mill Pond, Schoolhouse Pond, Seymour Pond, and Walker Pond. Those 4 ponds were also had postings at different times during the season. You can see the posting dates there. Any of the town ponds, other than Nickerson ponds, the accountability and responsibility for posting is with Amy Van Hoen and Chris Miller. She's the health agent. Chris is the Department of Natural Resources director. So they have the responsibility to make a posting for the pond, not APCC, not BPC. So I think what's also interesting when you look at this is that people will typically say, well, you know, when it gets really, really warm is when you'll see most of the postings or potential issues. But you can see we had a posting in May, June, July, August, and October. Pretty much every month from May through October, we saw a posting or a higher toxin level. So I think that's important to recognize as well, that these blooms can occur at not just in July and August. The posting is for a 2-week period following Mass Department of Health guidelines, EPA guidelines, WHO guidelines, until a lower toxin reading is seen. So we'll test more frequently from the date of the first posting until the 2-week period's over. Once we get a low reading, we clear the posting, or the town clears the posting. So next slide, Becca, please. So that was the year in 21. And again, that data and other pieces of information for any of the years, if you want to look at it, they'd be on the BPC website. Here is next slide, Cyanobacteria Screening for 2022. This particular piece, you can move ahead, Becca. Thank you. That particular, this particular piece will be on the clipboard for all of the 5 kits that you'll pick up at Gwen's garage. Gwen Pelletier was a 6-year board member at BPC, and she's allowed us to use her garage. She's been really great about that. So it's 1247 Long Pond Road, Route 137. She has a U-shaped driveway, so just to keep traffic flow going early in the morning as efficiently as possible. Just kind of, park on the outside of the U so cars can get around you and get through her driveway. Each of the kit includes all this. I don't have to necessarily read all of this, and again, this will be on the clipboard, but your kit will include everything you see here. Most importantly, well, it's all important, but there'll be 3 brown bottles, 500 milliliter bottles. They'll be labeled net for the netto sample gathered, WLW whole lake water sample gathered with the 1 meter tube, scum. If there is a scum that you see, you scoop up the scum with the water around it and label that scum, of course. There will be an additional 1 sample grab that's picked up. So the 3 brown bottles, 500 milliliter, they're pretty big. They're for the aforementioned 3, but the newest one will be this small glass grab bottle. And really, you're going to go into the pond and just scoop the surface water. I almost call it a surface grab at waist depth in the pond. And that'll be labeled grab. The tow net will be there, the turbidity tube or the disc will be in the kit, and the 1 meter tube for the whole lake water sample will also be in the kit. So next slide, please. Here is the data sheet. So my suggestion here, as you can see, is that you have 1 member with the clipboard who's filling in the data sheet, and it's basically self-explanatory. Becca and Kevin have made a few small edits to this year's form, but primarily, as I mentioned earlier, we'll be looking at gathering data on the weather, on the water, on the aquatic life, and any kind of human or pet activity you see. So I don't think you want me to go through each of these boxes, but maybe the more important ones that aren't self-explanatory would be the sample location coordinates, which is the 3rd box on the left-hand side going down. The rest of it above that's pretty simple and obvious. The coordinates I have a compass on my app, a compass app on my phone, which I think is fantastic. It'll give you the GPS coordinates. So you want to put the GPS coordinates in that area where we've identified it as sample location. The team name, numbers, photo. You should take a photo every time you go out of your site. And the photo you would send to cyano@apcc.org. So photo taken should always be yes. So any one of the 2 or 3 or 4 people on your team will have a phone. So please do that and send it to APCC. And then the information below that is this is all really important. So I'm not trying to go through this quickly, but much of it's self-explanatory, but really this is the devil in the details. So as citizen scientists, really pay attention, and whoever's got the clipboard, make sure you get a sort of a common agreement of the team as to wind speed, wind direction, rainfall conditions, all of the above. It's really important data. So I'm only going through it quickly because it's probably evident, but take your time and really get a team opinion on these things. And then the water assessment is, you can see waves, odor, if you smell anything, turbidity. I like to say that, well, how do you know if it's turbid if you're just looking at the pond? Well, you get a sense if it's turbid if you're using the turbidity tube, and you dunk that tube, you can get a good visual of whether the water is clear, turbid, or opaque, etc. Watercolor is pretty self-explanatory. In the Secchi depth reading here, I think what we want to do is on the far right-hand side, and I haven't talked to Kevin or Becca about this, but since 90% of our ponds will be tested using our turbidity tube, the Secchi tube, you'll dunk the Secchi tube and you'll get a meter reading on the tube. So I would suggest putting that in the far right box.
Yep. Yeah. Just to touch on that quick. Yeah, if you're Roy or anyone else taking a Secchi disc reading from the middle of the pond, then, you know, those first 3 boxes are, you know, you take a couple readings and then take the average and put the average in the box on the right. But if you're just taking a Secchi tube sample, you know, from shore, as Marty said, if it's the other 90% of the samples, then yeah, just put your reading in centimeters on that last average box on the right.
Right. And then next is the visible. If you see a visible scum or particulate, please take a photo of that, as I'd said earlier. But here you'd say yes, and you'd be sending the photo along to apcc.org. And then anything that you notice that you think is interesting, but it's always good to just catalog fish, birds, plants, anything that you see around the pond, and you'll start to get to know your pond really, really well if you don't already, and certainly the site that you're taking pictures from. And we like to see the changes in aquatic life and human and pet activity over the season. So there's the monitoring field data sheet, which is really, really important. So please take your time with that, and again, get a team consensus when you're filling it in. The clipboard person can be the person barking out the questions, and then people can come to a consensus. So next slide. So here are the actual physical sample grabs. WLW is whole lake water, and a grab sample. Both will be using the 500 milliliter, the 500 milliliter bottle. The brown plastic bottle will be for the whole lake water sample. And you can see a picture of that bottle right up top in the middle of the screen. And then use the small glass bottle. I don't have a picture of that, but it's a really small glass bottle, probably about yay high, a couple of inches tall. And it's glass, it's not plastic, it's copper colored, be pretty evident. You're gonna label that grab, and then you'd record on each of the bottles, whether it's a whole lake water, a grab, or the net tow. You'll record pond name, Sheep Pond, Brewster, June 1st, 8 a.m. So you'll label each of those bottles with those 4 important elements: pond, town, date, time. Before you collect the samples, rinse each of the bottles 3 times in the pond water, just to make sure the bottles will always be clean, but we're sort of conditioning the bottles to the pond specific. And collect a whole lake water sample at waist height. So go out to waist height and collect that sample using the 1 millimeter plastic clear tube without touching the sediment bottom. Collect the grab sample in the small glass bottle, again at waist height from the surface, almost like a kid would be dunking their bottle to fill up with pond water. You're just making a sample grab, again, from the waist high, out in the pond. Put the samples immediately in the kit near the ice pack, right on the ice pack, which will be in the bottom of every kit when you guys pick it up in the morning. Okay, next page. So here is the next sample, and it doesn't really matter to me. As a team, you'll get into some order that works for you, but the next sample that you'll take will be the net tow. And this is a bloom forming colony sample. It concentrates everything in the pond to the bottom of the cod end of the net. Is a plastic end of the net below that black stripe that we call the cod end. And you'll again use the same medium brown bottle, the 500 milliliter bottle, labeled again with all the specifics we talked about. Rinse that bottle first 3 times, dunk the net 3 times before you even start the process, and lock the bottom of the net. You'll see a little plastic, whatever we call it, lock at the bottom that will keep the water in the cod end. So walk into the pond around mid thigh or waist, and you can see there's our citizen science #1 volunteer, Chuck Medansky, throwing his net in Cliff Pond. So he's in about waist high, give or take. Throw it 3 meters horizontally, 3 meters being 10 feet, because you'll be throwing it standing up as opposed to Roy's team who will be in a boat. So it'll be a little bit different, but pull a net horizontally to make sure that the cod end has sunk so that you're gathering your samples in the cod end. Pull the net in slowly, hand over hand, and then rinse the side of the net by dipping it in and out after collecting the water. This doesn't come off as easy when you're reading it and saying it to someone, but when Becca shows you her sample video, it'll be pretty clear to you. And everyone who's out there will be with experienced people. So it just takes a little bit of fun learning how to throw it and tow it back in, but it's pretty simple. You pour the sample then from the cod end into the bottle. The cod end holds about 250 milliliters, I found out. So you will need to toss the net twice to fill up the 500 milliliter bottle. Pretty easy, but it'll take 2 tosses. And for the second toss, you're going to want to dunk the net, clean it 3 times again before you toss it. And then put that sample into the kit as well, near the ice. Okay, next slide. Step 5. And again, this entire piece that you're looking at here will be on your clipboard. So if you get out there for the first time and you forgot from last year or you're new, you can look at everything we're looking at right here. So now you're returning the kits and samples back to Gwen's garage at 1247 Long Pond. The net, whole lake water, and grab bottles are in the cooler. Put them in the cooler in the garage. There'll be a big, beer cooler kind of cooler so we can gather everyone's samples that same morning. Take your data sheet out that you filled out, place that in a folder that I have next to the cooler. So we have your samples and we have your written material on your pond. Leave the kit back on the table where you found it. This year we purchased, because we're growing so much and we wanted to make it easier for our friends over at APCC in the lab to have only one session and not have an 8 a.m. and a 10 a.m. session. All teams will be going out at 8 a.m. on Monday, Tuesday, or Wednesday, however you're assigned. So we had to purchase 2 additional kits to make it up to 5 kits. So we're able to put 5 teams out there at once each of those days. Before you're totally finished in the garage after the return is rinse and clean everything out. The net, the IT tubes, the Secchi tube. Hang the net up where you found it in the garage. We'll have hanging hooks that be pretty evident. And put the kits back in the same order that you found them. Anything you see that's unusual, you're going to please share that with me. You can email me, you can call me, you can text me. And each time you sample, please take again a picture of your site and of any scum, if it exists, and email that to cyano@apcc.org. Okay, next slide. So I'll turn it over to Becca.
Yeah, thank you, Marty. That was great. Hi, everyone. So the protocol that Marty just went through is approved by the EPA for the CMC Collaborative, and it was developed with help from our partners at UNH, Nancy Leland and Dr. Jim Haney, who Marty was talking about earlier. Like both of them said, we are taking new toxin test samples. So just a few notes on some of the stuff that Marty went over. If I can go back a few slides. You will have this 1 glass grab bottle. Marty, after I gave you those glass bottles, we did find out that the masking tape does slide right off of those bottles. So we've been using electrical tape. So after this week's sampling, we'll pre-label all of the glass bottles for you, so you don't have to worry about that. So if you have trouble with that, just make sure you let us know. Sorry about that.
No worries.
Yeah, also fair warning about that. On this field sheet, like Marty said, he pointed out all the good things. I just wanted to point out, I think you can see my cursor here. It says time of net sample. It's very important that you write the time of that you put the last bit into of the net sample into the bottle, not the time you get to the pond, because our net sample processing is time sensitive. So it's important that we know that. And at the bottom of this data sheet, it does say the email that we've been talking about. I did put it in the chat as well. Someone did ask about that. Also for collecting the grab sample, I don't know where that's written here. It is important that the grab sample in the glass bottle is taken about 12 inches under the surface and no closer than 9 inches to the bottom. So that's another good note to keep in mind when you're taking that glass sample. And also we do have to freeze those. Our bottles we give you are 125 milliliters. We ask that you please fill it no more than 3/4 of the way so that there is room for freezing. We don't want to break any bottles and lose a toxin sample. And then also with these photos, these are great examples. Please make sure to take a bunch of pictures of anything you see, and those can all be sent to us as well. And if you do see scum samples, just like Chuck did here, try to make sure we can see how big it is on the shore and things like that. So after you bring your samples to us, we'll go through and do a microscopy where we find the composition and dominance of the sample, which will then be used to kind of determine the results. Then we will freeze each of the types of samples that you take to lyse the cells, and then we'll run some fluorometry, which is taking pigments and collecting phycocyanin readings. Based on all of those results, we will be determining if it's important to send that sample off to the county for toxin testing. And if that happens, that does happen after all of the samples are processed. Give me just a second to pull up the training video. I'm just going to stop sharing my screen.
By the way, Becca, when you do share screen again, there will be there's a tab you can click on the top to share sound.
Okay.
Just make sure that it's activated.
Yeah, I think this is the this is the most recent one. All right. I'm just gonna play it from here.
Yep, I could hear the sound, by the way, so I think you should be all set.
All right, share screen. Can you see that?
Yep.
Yes.
Is there a way to? Okay, let's see if you can hear it.
2017.
Yep. Yeah, we can hear it.
Since 2017, the Association to Preserve Cape Cod Cyanobacteria Monitoring Program has relied on citizen scientists, volunteers to collect water samples across Cape Cod. The sampling process is simple and straightforward, but requires a keen attention to detail. Before we begin, thank you for your interest in assisting APCC with this critical program. Your help allows APCC to increase the number of ponds in our network, which improves our region's understanding of the cyanobacteria trends in our beloved freshwater ponds. When setting out to collect your samples, plan to be comfortably finished before 9:30 a.m. at the latest. Cyanobacteria bloom formations are more likely to be intact in the early morning before the water is disturbed. It is also important for APCC to obtain samples early in the day so sample processing can begin as soon as possible. APCC likely has planned a meat application for you and your group to pass off your samples to us, whether that be at our office in Dennis or perhaps the Barnstable Water Quality Lab in Barnstable. Please drop off your samples to APCC by 10 a.m. so the sample processing can begin on schedule. The sampling equipment that should be brought to each sampling event include 1 plankton net, 1 5-gallon bucket, 3 500 milliliter sample bottles per pond sampled that day, 1 glass toxin testing bottle for each pond sampled that day, 1 1-meter tube with a weight on the end, 1 digital thermometer, 1 clipboard with a pencil and the field data sheets, 1 cooler with frozen ice packs, rubber boots or waders, rubber gloves, a digital camera or cell phone with a camera, and any other personal equipment such as sunscreen, bug spray, hats, etc. Label the 500 milliliter bottles prior to heading to the ponds. 1 bottle should be labeled WLW, standing for Whole Lake Water, as well as the pond name, date of sample, and town. The second bottle should be labeled NET with the pond name, date of sample, and the town. The third bottle should be labeled scum sample with the pond name, date of sample, and town. Please also label the glass bottle should be labeled grab along with the pond name, date of sample, and town. It is important to note that if you see a visible cyanobacteria bloom or cyanobacteria scum on the surface of the pond, it has a chance of being toxic. Before you do anything, please take photographs of the pond and any bloom scum you may observe. It's good to take a photo before it is disturbed by any sampling. When taking samples, especially when a bloom or scum is observed, you should plan to wear gloves while sampling to avoid contact, and be sure to wash your equipment as soon as possible after sampling. If there is a visible bloom or scum, first take a few photos of the scum and email them to cyano@apcc.org. Then use the third plastic bottle to collect some of the scum sample. Again, this should be done with gloves on as to protect yourself. Collect the scum, making sure that no extra water will be added. Place the bottle in the cooler where it will be looked at under the microscope at APCC's lab to confirm the composition of the scum. Then take the glass bottle for toxin testing and face towards the center of the pond. Be sure not to touch the inside of the glass container when handling. Then please fill the container by dipping it into the pond surface until it fills to 3/4 of the way. Please be sure not to fill the bottle more than 3/4 of the way, as there needs to be room for freezing, and this is how much is needed for toxin testing in the lab. Place the bottle into the cooler on ice immediately. Then fill the whole lake water bottle with water and dump it. Do this 3 times in order to condition the bottle to the pond when you are sampling. Do the same procedure with the net bottle. The next sample to take is the whole lake water sample, WLW. Take the 1 meter tube with the weight on the end. Insert the tube into the water, making sure to avoid resting the tube end on the bottom of the pond. Place your thumb over the opening of the end of the tube that is above the water. You've now got your water sample. Place the weighted end of the tube over the sample bottle and empty the water into the sample bottle by removing your thumb. Repeat this until you have filled the bottle to at least 3/4 of the way. Replace the cover of the bottle and place it into the cooler where it will remain on ice until processed back at the lab. Time to collect the net sample. Just as you conditioned the net bottle at the start, the plankton net itself must be conditioned. Do this by opening the clip at the bottom of the net so the water can flow through the net uninterrupted. Dunk the net completely into the water and let the water run all the way out of the net 3 times. Close the clip at the bottom of the net and dunk the net into the water about halfway so that the net is filled with water. By doing this, a little bit of weight has been added, which will ensure that when you throw the net, it will land on the water the correct way. While holding the top of the net that is connected to the rope, throw the net horizontally across the surface of the water about 3 meters. When the net hits the water, begin pulling it towards you along the surface of the water at the rate of 1 foot per second. Once you have the net fully reeled in, dunk the net into the water to wash any cyanobacteria that may be clinging to the sides of the net down to the bottom of the net. Be sure not to reintroduce any new water through the top opening of the net. Hold the net vertically and wait for any excess water of the net to filter out through the net's mesh. Then insert the hose at the bottom of the net into the net bottle. Release the clip and the water sample will enter the bottle. Repeat this process in order to fill the bottle, but make sure you rinse the net 3 times between each toss of the net. Then place the sample bottle into the cooler to be processed at the lab. Be sure that the clip at the bottom of the net remains clipped when throwing the net, as if it falls off the net, the clip does not float and will be lost. Before leaving the pond, please make sure that you took a photo of what the pond looked like that day. Be sure to email any photos you take of the pond or scums to the APCC Cyano team using the email cyano@apcc.org. In this email, please include the pond name, town, and date of sample and photo in either the subject line or email body. It is important to have a photo of each pond in our records from each sampling event so that we can refer back to them when we get our results. So please be sure to take a photo every time you sample. Finally, bring your samples to your designated drop-off location with APCC by 10 a.m. APCC will then provide you with new sample bottles and data sheets for use at your next sampling event. When you return home, please be sure to rinse off your sampling equipment well and let everything air dry. Please do not leave your plankton net to dry in the sun, as long-term sunlight exposure can damage the net. Instead, hang it in the shade or in the shed or garage is ideal. Following the sampling process, APCC will inform your group leader of the results within 24 to 48 hours. Thank you for all your help in this important project, and we are excited to be able to work with you this season. Monitoring.
Let me just present it. Where's the button? There it goes.
All right. Yeah, Marty, want to take back over?
I will. The only addendum I would say to the that fun video and pretty effective video is that the BPC drop, the gathering of the kits and the dropping off of the samples all goes to Gwen's garage at 1247 Long Pond. So no one will be, other than myself or Ron Essick or someone else, if we're not available, none of us will be going to the labs in East Dennis. You'll just be dropping it back off at Gwen's garage. So can everyone see this page?
Mm-hmm.
Okay, good. So this is, just to finish up here, this is the team sample page slide for the season, and it identifies left to right, pond name, team members, contact information, schedule start date every other week beginning next week, and then the site for sampling, which maybe didn't show up on this particular, it's not wide enough to show up here to the right of schedule date, but you have it, and I'll send it around one more time to every one of the BPC volunteers. So for instance, Baker's Pond, team members, Burke, Wingard, Michaels. I put phone numbers for each of the team members, the schedule date. We start on Wednesday, so Wednesday would be June 1, and then 2 weeks later, June 15. 2 weeks later, June 29th, if I'm doing it right. So that's the way that will work, and it'll always work. You'll always be, your team will be the same group of people on the same day of the week. Every one of us goes out at 8 a.m. Now, I will plan on being, and Ron Essick will be working with me too when I'm not there, but I will plan to be at the garage at 7:30 a.m. So if any team wants to get out there a little bit earlier, and they want to get to the garage at 7:35 or 7:45, that's fine with me. I'll be there at the garage at 7:30. So I think other than that, I don't want to get in the middle of micromanaging the teams. So let's say Blueberry Pond, Jan McGann, Carol, Eric, and Amaya. There'll be a designated person who's sort of the team leader, if you will. I would guess that would be Jan and Carol, since they've been doing this for a long time. Actually, the entire team is pretty experienced. So you have phone numbers there, and perhaps maybe for the first sampling session or two, if you have new members on your team, it makes sense to maybe have the entire team there, so the new individuals can see the protocol and how it all works. And then maybe as you work throughout the season, I think it'll be obvious that you'll find that you only need 2 people, maybe 3. It's partly social, so you can do what the team wishes, but as you get rolling, you probably only need 2 people at the site at any given time. But having 3 or 4 or more people, Owl Pond has 5 people on their team this year. Having that many people on the team is a luxury, because inevitably there'll be a doctor's or a dentist appointment, et cetera. So just you guys manage your own team, stay in contact, and figure out who's going to be at each of the sessions, Monday, Tuesday, Wednesday, throughout the season. We start the season this year, all the Monday teams. So for instance, Elbow Pond, Greenland Pond, et cetera, et cetera. Any of the Monday teams have the day off. It's a vacation day. You won't be testing. It's Memorial Day. The lab won't be open, so there'd be no place for us to bring samples. So the Monday ponds will be tested later in the week by Becca and myself and others, but the Monday teams won't be testing for the first time until 2 Mondays from this Monday. The Tuesday ponds will start, like for instance, Blueberry Pond starts on Tuesday the 31st. Baker's Pond starts Wednesday, June 1st. It's just the Monday ponds that won't be in action in 2 days. So the end of the season will more than likely be at the end of October. And as we move into late September and October, sometimes individuals who might live somewhere else during the season might be leaving, but we'll work through that. I don't have a final date yet for the last sampling in October, but rest assured it'll be somewhere near the end of the month. I think the site for sampling is important too, particularly for people who've not done this before. The site for sampling is the fifth from the right-hand section on the page. And again, I'll send this entire finished page schedule out one more time to everyone in the BPC group. But I think that's pretty much it. I'm sure you might have some questions, but that's kind of the way it'll work. You can see the logistics slide here, which is every other Monday, Tuesday, and Wednesday, starting for us, starting Tuesday, the 31st of May. The sampling time takes about 20 minutes per pond. I think that's probably true. Most of us take a little bit longer, but but you'll find that once you get moving into the season, it just doesn't take all that much time. You'll have one person on the clipboard, one or two people in the water, and get the samples back. If you're picking them up by 7:45 or even 8 a.m. at Gwen's, undoubtedly you should be back to the garage by 9 or so, and you'll find that you'll be back even earlier as you get more experienced. And then I will take myself or Ron Essick, or if Ron and I are not available, I'll ask someone else for help. That we'll take all the samples from all of the teams to the APCC lab at the end of each morning. And that's, I think that's the last slide. I'm not sure.
Yep. Yeah, we can open up to some Q&A. Saw some questions from Chuck, but also just wanted to share Becca and I's contact information, our emails, of course, for some of the logistics, coordinate with Marty, but just in case, here are ourselves as well. Just to go into answer Chuck's recent question, do we take the toxin test and grab sample every time? Yes, for that. So our plan this year is, our options were either to sample every pond and then get our results, and then if the results are concerning enough, then we would go back out and take a toxin sample from each of those ponds that we found concerning results in. But given our, it's much better for us to have those toxin samples taken at the same time as the rest of our results to make sure everything lines up. We're choosing to have these toxin samples taken at every pond, and they'll only be sent on to the county if we find those concerning results. If we don't find those concerning results, then we'll just empty out the bottles, clean them out, and have them ready for the next sampling event. But yeah, of course, if there are concerning results, then we have those samples in our chest freezer, and we'll send them onto the county. Chuck, I can connect with you on cleaning. It is you do need to rinse each of the bottles. If you don't end up needing to send those bottles on to us to send to the county, then it's you defrost them, empty them out, and then it's 3 to 4 rinses of distilled water. And Marty and I talked about it, and I would say just arrange to pass those samples onto Marty, and then Marty could bring them to us, or you could drop off those toxin samples to us, or we could pick them up from you. However we want to do it. But yeah, we would arrange to collect those samples from you each time, to send on to the county as needed. And we're only going to send it on after you've completed your analysis for those ponds, if necessary. And also, how do I get those bottles back in time for the next sampling? When the bottles are dropped off to us, then we'll have fresh bottles, to replace the ones that have been dropped off. And yeah, and that'll, bring into the off week as well. And any other thoughts or logistics there, Chuck?
I think that's everything. I don't do any analysis on the grab sample. I need to get a copy of the new form. I don't have any of that. So if you could email that to me, that'd be great. And just for anyone else to know that, sometimes the water level in the ponds, like last summer, was very low, and you can't even get to hip height. And so if you're doing a whole lake water sample with an integrated tube, you could just record the height that you're actually taking from, right? Because it's not going to be 3 meters. It's going to, it's not going to be a meter deep. It's going to be less than that sometimes. So.
Yep. And just make sure to, if you're dealing with a pond where the water level is a bit lower, and make sure that you don't get any sediment in your samples, either the IT tube or the net sample either. This is the case. I remember, in elbow pond sometimes, as I'm kind of going through and kind of stirring up some sediment as I walk. Also make sure to cast your net in a different direction than, where you just walked through, because you don't want to get some of that sediment into your sample bottle. Just another thought I had coming off that.
Kevin, there's another question in the chat from Tom. He says, how important is it to use the sample tube versus just dipping the bottle in the whole lake for the whole lake water sample? Yep.
So it's part of our quality assurance plan to take our whole lake water samples with the IT tube. It's important to be consistent and adhere to specific equipment used for the quality assurance plan. It's using the tube as well, you get the profile of cyanobacteria in the water column from the top to the bottom, which is important. Just noting too that the difference between the whole lake water and the grab sample is that the whole lake water sample needs to be taken through the tube, but the grab sample, you don't use any equipment to take that sample. You just, as Becca said, wade out and take it 12 inches under the water. In the video, we will be tweaking the video to illustrate the 12-inch under the water and at least 9 inches from the bottom to collect that grab sample. It's not just from the surface, but you plunge it under and then bring it up and cap it, and make sure it's not too full, because we've already dealt with last week. We had a couple of those toxin sample bottles filled up a little bit too high, and we opened our freezer, and a couple of the glass bottles had broken, and we had to go through and clean everything out. So yeah, make sure they're not filled too highly either. And I see also here, do we pre-label the bottles at the garage except for the net? I would encourage pre-labeling, all your bottles before you go out to save your time. If for taking down the time that the net sample was taken, as Becca said, make sure to wait to mark that down until you're in the field and you've taken that sample, so we know, because it's important for us to know that, say, even if you went out there to take the samples at 8 a.m., if you didn't take the net sample until, say, 8:20, then that 20-minute difference is important to us, as some of our procedures are pretty time sensitive, as Becca said.
Can I ask a question?
Oh yeah.
So Becca, in your video, when you took the grab sample in the glass jar, I don't think I saw you conditioning the jar by dipping it in the water and rinsing it 3 times, but we've all been trained to do that with the brown plastic containers. So am I understanding correctly that one does not precondition the glass grab bottle?
Yes. Conditioned plastic, not conditioned glass. That might be an
Okay.
easy way to remember it.
Thank you.
And just the note on that too is it is a different protocol that we're using for collecting the toxin bottles. It's a EPA and state protocol. And so we just adhere to that, and it doesn't specify needing to condition the bottles.
Good, thank you. And second question, my team happens to be one of the teams where at Seymour Pond that falls on Monday, Memorial Day.
Yep.
And so we get a holiday. And then damned if it doesn't work out that we fall again on Monday, Labor Day. They're
Okay.
aligned every other week. And so once again, will we be having a holiday on Labor Day because APCC is closed, or is that being handled differently?
Yeah, Becca, do you want to answer how we're playing around holidays?
Yep. So our holiday sampling schedule for those Monday holidays, I believe all of our holidays this season are Monday holidays.
Yeah.
We are moving all Monday sampling to that Tuesday, and our APCC staff can go out and take those samples for you guys since you only have 5 kits and we need our samples by 10.
Okay.
So
All
yeah,
right.
so Ann, Seymour, and then any of the other for this coming Memorial Day, anybody who was originally scheduled for that Monday, you have the day off, and Becca or I will go out and collect each of those samples that were supposed to be taken on Monday on Tuesday morning, and then Marty will meet us up with the samples that were scheduled for Tuesday, and that you all collected on Tuesday. So
Got it. And the same drill on Labor Day. That was really my
Exactly.
question.
Yes.
Good,
Yep.
thank you.
Yep. Mhm. All right, anybody else? And of course, feel free to, connect with me or Marty or Becca as well if you have any other questions or comments or want to discuss anything else.
I'm going to stop the recording now.